Hello, everyone. It is my pleasure to introduce the study single-cell sequencing reveals the role of IL-33 endothelial subsets in promoting early gastric cancer progression. Gastric cancer remains one of the most lethal cancers worldwide due to its rapid progression and treatment resistance. Stromal cells and cytokines in the tumor microenvironment are involved in the processes of tumors. Our highlights are as follows. First, we comprehensive single-cell analysis of EGC. Second, we found CH25H CD4 T-cell subclusters are specifically abundant in EGC. Third, IL-33 and AMAMTSL2 endothelial cell subpopulations increased in EGC. Fourth, we revealed that IL-33 endothelial cells promote angiogenesis and growth of EGC by regulating adhesion molecule expression. In this study, we harvest clinic samples. Then construct a single-cell atlas underlying the cellular and molecular characteristics of the TME across different lesions. We subsequently generate patient-derived EGC and AGC organoids and reveal that endothelial-derived IL-33 promote the growth of GC. 8 major clusters and 22 subpopulations were finally identified. The proportion of epithelial cells decreased gradually from gastritis to AGC. However, the proportions of T&NK cells, B cells, plasma cells, fibroblasts, myeloid cells, and endothelial cells increased following the progression of GC. We identified 11 subclusters of epithelial cells, and found one cluster as the cancer-free cells, which had 7 distinct lineages. Among them, curve 1 and 2 differentiation trajectories cover the lineage changes from CAGIM to AGC. It was also found that genes such as OLFM4 were significantly increased in EGC and predicted a poor prognosis for patients. Further subclustering analyses revealed 8 CD4 T-cell clusters. Among them, the proportions of TREG and CCR7 naive increased in GC, whereas CH25H CD4 T-cells were significantly enriched in EGC. Notably, we identified the understudied CH25H CD4 subpopulation. The expression of CH25H was positively correlated with CD4 T-cells and CR-naive T-cells. Survival analysis also revealed that high CH25H expression was correlated with poor prognosis in patients with EGC. Among CD8 T cells, we identified 10 clusters. We found that the proportions of CD8 TEM and GFPT2 CD8 T cells increased, whereas the proportion of MAI T cells decreased with the occurrence of GC. We divided fibroblasts into 8 subclusters. The CA-FINFLA PRSS35, CA-FINFLA MFAP5, and KFM KRT19 subclusters were enriched in AGC. PRSS35 and MFAP5 were specifically expressed in CAFs and associated with poor prognosis in GC patients. Then, we identified 7 subclusters in endothelial cell. The relative IL-33 Venous-1 and AMAMTSL2 Artery-2 proportions gradually increased, whereas CA4 capillary-2 dramatically decreased with GC progression. We revealed that high expression of IL-33 was correlated with poor patient prognosis. Zhou and EGG analyses showed that the marker genes of the subcluster were enriched in cell adhesion pathways. We also found that the expression of IL-33 in EGC samples was high. Then, experiments revealed that the overexpression of IL-33 increased cell tube formation ability. Furthermore, we found that IL-33 promoted the growth of ECs and reduced apoptosis. IL-33 from ECs facilitated the growth of GC organoids. We then tested the function of IL-33 endothelial cells by co-injection of organoids and ECs into mice. The results revealed that ECs increased the tumor formation capacity and IL-33 overexpressing ECs further promoted the growth of GC organoids. Subcutaneous ultrasound experiments in mice also support the above conclusion. Taken together, these findings indicate that IL-33 endothelial cells promote the growth of GC organoids. We subsequently used transcriptomic and Western blot analysis to elucidate the mechanism of IL-33 in endothelial cells and EGC organoids. These results confirm that IL-33 facilitates angiogenesis by upregulating PECAM1 and CD34. We also found that the EGC with high expression of IL-33 and CD34 had deeper invasion and higher pathological malignancy in clinic EGC samples. In summary, as one of the most comprehensive single-cell sequencing studies to date for EGC, our study provides a unique resource for generating novel biological insights into tumor cell types, subtype-based TME compositions, and cell-cell interactions in EGC. Mechanistically, IL-33 enhances the survival and angiogenesis of endothelial cells by upregulating the adhesion proteins PECAM1 and CD34. Endothelial-derived IL-33 could also promote the growth of EGC organoids through increasing KRT17 expression. Notably, we also found that high expression of IL-33 was positively correlated with the depth of invasion and malignancy of EGC in the clinic.